Hey everyone. So I am telling you that my I have filled a form of GATE exam, and GATE exam will be held in February month on 6th, 7th and 13 and 14 uh, date of that month. So I filled that form in biotechnology and life science. So I'm going to study for that exam. Uh, so uh, now I'm going to start a series of my studying gate where I will be studying biotechnology and life science subjects, and we'll first discuss the syllabus of that subject. Hey everyone, so here is the syllabus of GATE Biotechnology. So before that we move into the discussing the topics, we must understand uh, why we are giving GATE. So GATE can be given by the engineering students uh, pass out of BTech BE exams. Um, Once they get graduate, or maybe they are in the final exams of graduation of engineering, they can attempt this exam. They can go for this exam. So what are the benefits of this exam? First of all, this exam is going to uh, make you understand that how knowledgeable you are, whatever you have studied in your graduation program. Next is you will get the opportunity, if you got a good score in this, you can go for a master's degree, you can go for a PhD degree. There are many fellowships which are um, given by the um, government institutions who actually got a very high ranking in GATE score. So if you want to have a good career after graduation in engineering, you must go for this exam. You can go for this exam, otherwise you can go for other options also. So here is the syllabus. First is the biotechnology, and first section will contain engineering mathematics. So it will linear algebra, calculus, differential equations, probability and statistics, numerical methods. Then we will go for general biology. Uh, Here we will go for, you can see, there's biochemistry, microbiology, immunology. So this comes in the general biology section. Then we will go for genetics, cellular and molecular biology. In genetics and in evolutionary, you will get Mendelian genetics, where you will understand Mendelian inheritance, gene interaction, complementation. Then we have chromosome mapping, extrachromosomal inheritance, microbial genetics, transformation, genetic disorders, population genetics. In cell biology, you will understand the structure, the growth of the cell communication. You have to read all these topics. Before This is just a reference to the syllabus, but if you want to understand that which topics are important, you must attempt the earlier previous papers of GATE, then you will get an idea of how which topic is more important and which questions you have to focus on. Okay, this is then we have fundamental biology engineering, then in which we have to understand the bioenergetics, the and transport processes and some engineering principles which can be applied to the biological systems like stoichiometry, recycle, bypass, all these. So this actually a kind of bioprocess. We study this in bioprocess. Then here is the bioprocess engineering and process biotechnology, where we will understand about the bioreactors, upstream and downstream processing, and the instrumentation and process control. Then plant, animal, and microbial biotechnology. So microbiology here it is. Then we have recombinant DNA technology and the tools which are used in biotechnology, like molecular tools are there, analytical tools like DNA fingerprinting, RAPD, RFLP, IR, fluorescence, FTIR, MS, NMR, computational tools. So the syllabus is vast, but you have to choose that which topic are important and which topic you should skip. Then, if I talk about the subject life sciences, so in life science, you will get five sections, and from those five sections, you have to attempt only two sections. No, actually, you will get if you are going to attempt the life science exam in GATE, then you have to compulsory attempt the chemistry part, and other five you have to choose one subject. The other five are biochemistry, botany, microbiology, zoology, and food technology. I am a student of biotechnology, so I am going to focus on microbiology and food technology and biochemistry, and among them I will choose only one subject, that which subject I like to attempt in that exam. So here chemistry, we can see structure and bonding, atomic structure and periodicity, S, P and D block, elements, chemical equilibria, electrochemistry, reaction kinetics, thermodynamics, structure, reactivity and correlations, and organic reaction mechanism. So you have 8 sections in it. Okay, next is our what is there in biochemistry. So section 1, section 2 and section 3, there are 6 sections. Okay, in first section we have the biomolecules like uh, amino acids, carbohydrates, lipids, water, nucleic acid, protein structure folding, and different proteins are here like hemoglobin, myoglobin, lysozyme, ribonuclease A. So we have to study all these. Then we have section 2, enzyme kinetics. Enzyme kinetics, um, glycoly, glycolysis, TCA cycle, pentose phosphate, all the metabolism is in the section 2. Then section 3 will have separation techniques, all the chromatography, spectroscopy, mass spectroscopy, metry, and how you will characterize the biomolecules, different techniques of characterization of biomolecules. Then we have cell structure and organelles, biological membranes, transport across membrane, signal transduction, hormones and neurotransmitter, DNA replication, transcription and translation, biochemical regulation of gene expression, recombinant DNA technology and applications, PCR, site-directed mutagenesis, and DNA microarray. These are also techniques. and here which are actually the DNA biology, all are in the section 5. Then we have section 6, which is immunology part, active and passive immunity, complement system, antibody structure, cells of immunity. All this is the part of immunology. And then we have immunological techniques like immunodiffusion, immunoelectrophoresis, RIA, and ELISA. So we have to complete all this. We'll go for microbiology. Let's see what is there in microbiology. So there's a historical perspective, that how the history of microbiology, how it, what who started the study and all. Methods in microbiology, like what methods we use. Then we have microbial taxonomy and diversity, prokaryotic and eukaryotic cells, microbial growth, control of microorganisms, microbial metabolism, microbial diseases and host pathogen interaction, chemotherapy, antibiotics, microbial genetics. Then we have microbial ecology. So there are 10 sections in microbiology. Then food technology. So food technology, we have first is food chemistry and nutrition. Here we will study about carbohydrate, protein, lipids, pigments, food flavors, nutrition, balanced diet, enzymes, chemical and biochemical, C changes. Okay. Then we have food microbiology, microbial growth, toxins from microbes and fermented foods and beverages. Then we have characteristics of microbiology, food products and technology, processing principles, food pack aging and storage, cereal processing and products, oil processing, vegetable processing, fruit and vegetable processing. Then we have plantation crops processing, milk and milk product processing, processing of animal products, waste utilization. Then we have food standards and quality, where we have to study about FPO, PFA, EGMOC, HACCP, food plant sanitation and cleaning in place. Then we have food engineering, mass and energy balance, momentum transfer, mechanical operations, thermal operations, and mass transfer operations. So these are the top subjects which are my interest, and hopefully, uh, so from today we are going to start studying, and a series we are going to start where we will study for 2 hours per subject, like biotechnology, biochemistry, chemistry, and food technology. So 2 hours per subject.